Hi everybody, my name is Michaela Harrison and I am going to be talking about sickle cell anemia, which is what I have researched for the last 8 weeks. So let's dive in. First, I want to start off by answering the question, what is sickle cell anemia? Sickle cell anemia is also known as S, disease. It is an autosomal recessive disorder, and the disease affects the hemoglobin in red blood cells, or RBCs. The official name of the gene responsible for this disorder is Homo sapiens mutant beta globin, or HBB gene. The name of the disease comes from the shape of the cell. There is a difference in shape, as you can see in the diagram, between a healthy red blood cell and a sickle cell anemia red blood cell. So let's talk a little bit more about the structure, the organization, and the location of eukaryotic chromosomes now. So again, the structure, they are either sickle or crescent in shape. They're stiff, they're not concave, so they don't carry oxygen as well as a healthy red blood cell, as you can see in the middle diagram. And the shape and structure often lead to clotting due to such stiff hemoglobin molecules inside of the sickle cell, whereas in the regular healthy red blood cell, they are very, very flexible and not so rigid. So the organization, so the nucleic acids in a healthy red blood cell are different than the nucleic acids in a sickle red blood cell, or their organization of those nucleic acids are switched. So in a healthy red blood cell, we get a different protein than we get from a sickled red blood cell. The location is 11p15.4, and I'm going to go over the diagram on the far right a little more in depth later on in my presentation. So the next 4 slides that I'm going to cover are from Project 2 that we did, with the Punnett squares, talking about parents, genotypes, as well as offsprings, phenotypes for offsprings and for parents, and then the probability that the offsprings will be afflicted with the disorder. So the genotype for an afflicted person with sickle cell is HB capital S, and the genotype for an unaffected person is HB capital A. For someone with a sickle cell trait, they will have a genotype of HB capital S capital A. So let's run through these 4 scenarios. First, both parents are afflicted with this disorder. So the parent's genotype will be HB capital SS, as will their offspring, because they are both affected with the genetic disorder. Both the parent and the offspring will show sickle cell disease, and the probability that the offspring will be afflicted with the disorder is 100%. All right, possible scenario number 2. One parent is afflicted with the genetic disorder and one parent is unaffected and not a carrier. So the 2 genotypes of the parents will be HB capital S and HB capital A. So the genotype of the offspring here is HB capital S capital A. The phenotype, one parent has the disease, the other parent is unaffected, and the offspring's possible phenotypes are that they will have sickle cell trait. So they do have a 0% chance of actually being afflicted with sickle cell disease, but they do have a 100% chance of being afflicted with sickle cell trait. So third, the parents, one parent is afflicted with a genetic disorder and one parent is considered to be a carrier. The genotypes HBSS and HBSA for the parents will be the same for the offspring, and the phenotype of the parent and the offspring will be the same, where it will be sickle cell disease and sickle cell trait. So the probability that the offspring will be afflicted with the disorder is 1/2, or 50%. And all the Punnett squares you see to the side are Punnett squares that I made for this project, and so if you need a visual, I hope you will pause the video and stop and look at them, and yeah. So the last scenario I want to cover, neither parent is afflicted with the genetic disorder. Also, neither of them are carriers, so their genotype is HBAA, as will the offsprings be. The offspring and the parent will have the same phenotype of no sickle cell anemia or unaffected, and the probability that offspring will be afflicted with the disorder is 0%. So now let's talk about normal chromosome number and behavior in human cells. So this can be found on chromosome 11, and its behavior in human cells is that the disease affects hemoglobin in red blood cells, as previously stated. Just to go in depth in that, hemoglobin molecules are responsible for carrying oxygen and transferring it throughout the body. So I just threw in these 2 charts on the bottom left, in the top right. They are basically saying the same thing. It just gives you a different section of the red blood cell cut open so you can just see inside of it. I like the one on the top right because it actually shows how the sickle cells can block veins or arteries. But in the bottom left picture, you can see the rigid structure inside of sickle cell red blood cells, with the hemoglobin being very, very stiff, whereas in a normal red blood cell, the hemoglobin molecules are round and very, very flexible. So symptoms of sickle cell anemia, this is also part of how it affects our bodies. Jaundice, low levels of oxygen, weakness, fatigue, and dactylitis. So some people exhibit physical effects of this disease early on in life, while others may begin experiencing them later on. Chronic complications in a person with sickle cell disease can lead to mortality. Signs, symptoms, and severity will vary from person to person. So this is the pedigree chart that I created for one of our projects. Feel free to pause the video and look at it in detail if you'd like. The key at the top shows that people with sickle cell anemia in this family will have a blue square in the top right-hand corner of either their square or circle, and people with sickle cell trait will have a pink circle in the middle of their square or circle. Obviously, unaffected people won't have anything inside of their square or circle. So my hypothesis about the underlying inheritance patterns and phenotypes of future generations from my pedigree, so as you can see in the chart, 18 out of 25 of the family members have sickle cell disease or sickle cell trait. My hypothesis is that any afflicted member, disease, or trait that marries a person who is also afflicted has a high probability of passing down either the disease or trait. It seems like the best way to ensure a couple's future child would not be affected with sickle cell anemia, the disease, would be that they have a child with someone who is not even affected by the trait. So here are two examples from my pedigree. Jared could marry anyone and ensure that his child would not have sickle cell disease, since he doesn't even possess the trait. However, Michelle would have to marry someone who is unaffected to ensure her children would not get the disease, since she does have sickle cell trait. Any member of the family who has sickle cell disease should marry someone who is unaffected to ensure their future children would not also have the disease. In this case, they still run the risk of passing down sickle cell trait to their offspring. If afflicted members choose to marry someone who is also afflicted with either the disease or the trait, they run a higher risk of their children inheriting the disease. These children will most certainly inherit at least the trait. So the molecular process of gene expression from DNA to protein. I briefly touched on this earlier in my presentation, but I want to go into more depth here. So in DNA of someone with sickle cell, the middle pair of nucleic acids are flipped. In a healthy red blood cell, the middle pair has adenine on the top and thymine on the bottom, but in someone with sickle cell, they have thymine on the top and adenine is on the bottom. This leads to two different proteins being formed. So glutamic acid, or GLU, is formed in a healthy red blood cell, where valine, or VAL, is formed in a sickled red blood cell. Mutations. So mutations include beta thalassemia. Beta thalassemia is a blood disorder that reduces the production of hemoglobin. Most of the mutations involve a change in single DNA building block or nucleotide within or near the HBB gene. Other mutations insert or delete small numbers of nucleotides in the HBB gene. This specifically results in a condition called beta plus thalassemia, and a lack of hemoglobin disrupts the normal development of red blood cells. This could lead to a shortage of mature red blood cells, which would result in insufficient transportation of oxygen. With a lack of oxygen flow in the body, tissues can lead to poor growth, organ damage, and other health problems associated with this. So techniques used in genetic screening and testing. So there are 94 genetic tests available for this to test for this disorder, but the most common is a complete blood count, or a CBC. They check for hemoglobin S, which is the defective form of hemoglobin that underlies sickle cell anemia. There are multiple ways to draw blood depending on the person's age. The blood will get sent to the lab where it is then tested. Negative screening results mean that someone does not have the hemoglobin S issue, but positive screening results will result in further testing to confirm that sickle cell is present. Other tests include hemoglobin electrophoresis test, high-performance liquid chromatography, or HPLC test, and a DNA test. And these are my references. I hope that you have enjoyed my presentation and learned something new about sickle cell anemia. Best of luck to everybody on their final.